Welcome to the deep dive. Today we're diving deep into, well, something that seems simple but is actually an architectural marvel, the bacterial cell. We're going to unpack the structures these tiny organisms use to thrive, survive, and sometimes, yes, cause infections. And let's jump right in with a really striking example. Forget static blobs under a microscope. Think about Neisseria gonorrhea. The cause of gonorrhea. It uses these hair-like things called pili for sticking to surfaces and also for this weird kind of movement called twitching motility. But here's the kicker. Seminal fluid actually cranks up this twitching, makes the pili bundle spread out into single filament.
Which makes them way better at moving and sticking. It's a really effective mechanism for infection. And that really sets up what we want to do today. Connect these structures directly to what microbes actually do. Even these supposedly simple cells have incredibly sophisticated behaviors.
Yes, simple is a loaded word here. And speaking of words, before we dig into the architecture, we should probably touch on the term prokaryote. There's a bit of a debate there, isn't there?
Oh, absolutely. Historically, folks like Stanier and Van Niel, back in the 60s, they defined prokaryotes by what they didn't have. No nucleus wrapped in a membrane, no complex internal organelles,
Defined by absence.
Exactly. But, well, modern science, especially genomics and biochemistry, shows that bacteria and archaea are fundamentally different domains of life, really distinct evolutionary paths.
Right. So people like Norman Pace are arguing we should just, ditch the term prokaryote altogether. It lumps two very different groups together based on lacking something, rather than highlighting their unique innovations.
That's a fair point. It masks a lot of biological diversity.
Okay, so let's start our tour from the outside in. Morphology. What do bacteria actually look like? Most people probably picture spheres, cocci, or maybe rods, bacilli.
Those are the most common, definitely. But how they arrange themselves is often key for identifying them. Cocci can be in pairs, diplococci, or chains like streptococci, or even those grape-like clusters you see with staphylococci. Rods can form chains, too.
And then you get the less standard shapes. Vibrios, which are sort of comma-shaped, spirilla rigid spirals, and spirochetes, which are flexible spirals and often, pathogenic. They move in a really unique way because of where their flagella are.
It's not just shape, though. The size range is pretty staggering. Your average E. coli is tiny, maybe a micrometer or two long. But then you have mycoplasma, super small at 0.3 micrometers. And at the other end, Epilepisium fischelsoni.
That's the giant one, right?
It's enormous, up to 600 micrometers long. That's bigger than some eukaryotic cells like paramecia.
Okay, that immediately brings up a fundamental problem. We're always taught that being small is good for bacteria because it maximizes the surface area to volume ratio, the SV ratio. Why is that so important?
It's absolutely critical. Think about it. Bacteria need to get nutrients in and waste out across their surface, their membrane. A higher surface area relative to the internal volume means more efficient exchange. Faster nutrient uptake means faster growth. It's a key driver of bacterial life strategy.
So, how does that massive epilepisium manage? 600 micrometers long, that SV ratio must be terrible.
It would be, except it has a trick. Its plasma membrane isn't just a smooth boundary. It's incredibly folded, convoluted, creating this huge internal surface area. So it gets around the volume constraint by just making more membrane surface available for transport, structure adapting to function, basically.
Clever. That's a perfect lead-in to the membrane itself. So the cell envelope, that's the plasma membrane plus anything outside it. But the plasma membrane is ground zero, right? The essential layer.
Absolutely essential. It defines the boundary of the living cell. It's selectively permeable, controlling what gets in and out. Plus, in bacteria, it's where a lot of the metabolic action happens, respiration, photosynthesis in some, building lipids, cell wall components. It's a busy place.
And the model we use is the fluid mosaic model. Lipids floating around, proteins embedded. What makes a bacterial membrane distinct, though? Is it just phospholipids and proteins?
Well, mostly. But they have something eukaryotes generally don't. Hopanoids. These are steroid-like molecules, structurally similar to cholesterol. They insert into the membrane and regulate its fluidity, make it a bit more rigid. And they're also thought to help organize proteins into specific functional patches, kind of like microdomains.
Okay, so we have this dynamic controlled barrier. How does it actually feed the cell? What do bacteria need? We talk about macroelements and micronutrients.
Right, macroelements are the big ones needed in larger amounts. Carbon, nitrogen, oxygen, phosphorus, sulfur, potassium, magnesium, calcium, iron. They make up the bulk of the cell's structures.
And micronutrients.
Micronutrients or trace elements are needed in much smaller quantities. Think manganese, zinc, cobalt, molybdenum, nickel, copper. They're usually essential as cofactors for enzymes, helping them do their jobs.
Got it. Now, getting these things across the membrane, especially when nutrients might be scarce outside, bacteria can't just rely on things drifting in, can they? What are the main transport strategies?
No, passive diffusion is pretty limited. Only small, uncharged things like oxygen, CO2, water can really move across easily just following the concentration gradient. It's slow and depends entirely on having more outside than inside.
So, strategy number 2 is facilitated diffusion, using protein helpers.
Exactly. These are channel proteins or carrier proteins that bind to a specific molecule and help it cross the membrane, still moving down its concentration gradient. It's faster than simple diffusion, but it has a limit. The carriers can get saturated. And crucially, it still doesn't work if the concentration inside is higher than outside.
Which is often the case if the cell is trying to accumulate nutrients.
Precisely, which is why most bacteria heavily rely on active transport. This is the key. Moving substances against their concentration gradient, from low concentration outside to high concentration inside. And that requires energy.
Okay, energy. How do they pay for it? I know there's primary and secondary active transport.
Right. Primary active transport uses energy directly from ATP hydrolysis. The classic examples are ABC transporters, ATP binding cassette transporters. They bind ADP, break it down, and use that energy burst to push a specific molecule across the membrane, usually just one type of molecule, so they're uniporters.
Okay, direct ATP power. What about secondary active transport?
This is a bit more indirect, maybe more clever. It uses energy that's already stored in the form of an ion gradient across the membrane, typically protons, the proton motive force, or PMF. It's like using a dammed up reservoir of ions.
So it couples the movement of the desired nutrient to the movement of an ion down its gradient.
Exactly. You have symporters, where the ion, like a proton, and the nutrient move in the same direction. The famous example is the lactose permese in E. coli, bringing in lactose along with the proton. Then you have antiporters, where the ion moves in one direction and the nutrient moves in the opposite direction across the membrane.
Keeping the charge balanced, maybe?
Often, yes. It's all about harnessing that pre-existing gradient energy.
And the fourth way, which always seemed really unique to me, is group translocation. How's that different?
It's fundamentally different because the substance being transported is chemically modified during the transport process.
Yeah.
The best example is the PTS, the phosphotransferase system. As a sugar molecule comes across the membrane, it gets phosphorylated. A phosphate group gets attached.
And that uses PEP, not ATP, right? Phosphonol pyruvate.
Correct. It uses PEP as the phosphate donor. This modification does two things. It traps the sugar inside the cell because the modified form can't leave via the same transporter, and it keeps the internal concentration of the original sugar low, helping maintain the gradient for more uptake. It's very efficient for grabbing sugars.
That is clever. Okay, one specific challenge, iron. It's a macro element, essential, but ferric iron, F3+, is notoriously insoluble at neutral pH. How do bacteria grab enough iron?
Yeah, iron scarcity is a major hurdle.
Yeah.
They solve it with siderophores, literally means iron bearers. These are small molecules the bacteria make in secret. They have an incredibly high affinity for ferric iron, binding it very tightly even when it's scarce or insoluble.
So they send these out to scavenge.
Exactly. The siderophore iron complex then binds to specific receptors on the bacterial surface and gets transported inside, often using, you guessed it, an ABC transporter system that uses ATP. It's a whole dedicated import system just for iron.
Shows how vital iron is. All right, moving outward from the plasma membrane, we hit the cell wall. This seems critical for shape protection, especially against osmotic lysis, right? Stopping the cell from bursting in dilute environments.
Absolutely vital. And the foundational material for almost all bacterial cell walls is peptidoglycan, sometimes called murine. It's like a giant mesh-like bag made of sugar derivatives. And acetylglucosamine, NAG, and N-acetylmuramic acid, NAM, linked together in long chains.
And these chains are cross-linked.
Yes, short chains of amino acids, stem peptides, hang off the NAM sugars, and these peptides are cross-linked to connect the different glycan strands, forming that strong continuous sacculus or bag around the cell.
And there's a neat trick in those peptides, isn't there? Something about D-amino acids?
Ah, yes. Most proteins in nature use L-amino acids. But the peptidoglycan stem peptides often contain unusual D-amino acids, like D-alanine and D-glutamate. This is ingenious because most proteases, enzymes that break down proteins, only recognize L-amino acids. So using D-amino acids makes the peptidoglycan resistant to degradation by common enzymes.
A built-in defense. Now, this peptidoglycan layer is the basis for the biggest division in the bacterial world, gram-positive versus gram-negative.
It really is. The Gram stain, developed over a century ago, still works because it reflects this fundamental difference in cell wall architecture.
So walk us through a typical gram-positive cell wall.
Okay, gram-positive. Think thick. A really substantial layer of peptidoglycan, maybe 20 to 80 nanometers thick. Embedded within this thick wall are teichoic acids. These are polymers, usually of glycerol phosphate or ribitylphosphate, and they have a negative charge. They help stabilize the wall and might anchor it to the plasma membrane below. The space between the wall and the membrane, the periplasmic space, is usually quite narrow.
Okay, thick wall, teichoic acids. Now, contrast that with gram-negative.
Gram-negative architecture is more complex, even though the peptidoglycan layer itself is much thinner, only about 2 to 7 nanometers. This thin layer sits within a much larger periplasmic space, which can be like 20–40% of the total cell volume. It's full of enzymes and proteins. But the real defining feature is the outer membrane.
An extra membrane layer outside the peptidoglycan?
Exactly. It's a lipid bilayer, but it's asymmetric. The inner leaflet is phospholipid, but the outer leaflet is primarily made of a unique molecule found only in gram-negatives, lipopolysaccharide, or LPS. This outer membrane is anchored to the peptidoglycan below by bronze lipoprotein.
LPS, that's a really important molecule, especially medically. Let's break it down. Lipid A, the core, the O side chain.
Right. Lipid A is the part embedded in the outer membrane. It's a glycolipid, and crucially, it's the endotoxin component. If large amounts get into the bloodstream, it triggers a massive inflammatory response, septic shock. Very dangerous.
Okay, that's lipid A. Then the core.
A core polysaccharide links lipid A to the outermost part, the O side chain or O antigen. This O side chain is a repeating chain of sugars, and it's highly variable between different bacterial strains. It sticks out from the surface, and it's often what the host immune system recognizes in the targets. Its variability helps bacteria evade immunity.
So lipid A is the toxic anchor, O antigen is the variable flag sticking out, and the outer membrane also needs channels, right? How do things get through it?
Good point. It has porins. These are typically trimeric proteins that form water-filled channels through the outer membrane, allowing small hydrophilic molecules, nutrients, waste products to pass through. They provide some selectivity based on size and charge.
Okay, so thick wall versus thin wall, plus outer membranal LPS. How does the gram stain physically work then? Why does the purple dye stick to one and wash out of the other?
It comes down to the decolorization step, usually with alcohol or acetone. In gram-positives, the alcohol dehydrates the thick peptidoglycan layer, shrinking the pores in the meshwork. This traps the large crystal violet iodine complex inside. It stays purple.
Right.
In gram-negatives, the alcohol acts as a solvent. It dissolves lipids in the outer membrane and easily passes through the very thin peptidoglycan layer, which doesn't shrink enough to trap the dye. So the crystal violet iodine complex washes out. Then, when you add the counterstain, usually pink safranin, the decolorized gram-negative cells pick it up and appear pink or red.
Makes perfect sense based on the structures. Finally, beyond the wall itself, some bacteria have even more external layers, like capsules or S layers.
Yes. Capsules are usually well-organized layers, often made of polysaccharides, sometimes protein. Think Streptococcus pneumonia or Bacillus anthracis. They're important for protection against drying out, but also, critically, for evading phagocytosis by immune cells. They make the bacteria slippery and hard to engulf.
And S layers.
S layers are different. They're highly ordered, almost crystalline arrays of protein or glycoprotein that self-assemble on the very outer surface of the cell wall in gram-positives, or the outer membrane in gram-negatives. They seem to provide general protection against environmental stress, osmotic pressure, enzymes, maybe even predation. They're very uniform structures.
Fascinating. Okay, let's move inside the cell now into the cytoplasm. We often hear about macromolecular crowding. What does that mean for the inside of a bacterium?
It means the cytoplasm isn't just a watery soup. It's incredibly packed with proteins, ribosomes, DNA, metabolites. It's very viscous, almost gel-like. This density affects how molecules move around, how fast reactions can happen. It also begs the question, how does anything stay organized in there?
Yeah, for a long time the view was kind of bag of enzymes, wasn't it? But that's changed with the discovery of the bacterial cytoskeleton.
Completely changed. We now know bacteria have intricate internal protein filaments that act as a scaffold, analogous to the eukaryotic cytoskeleton. It's crucial for shape, division, organization.
So what are the key players? I know FTZ is a big one.
Right, FTZ is a homologue of eukaryotic tubulin. It forms a ring, the Z ring, precisely at the middle of the cell. This ring constricts during cell division, guiding the synthesis of the new cell wall septum that divides the two daughter cells. Without FTSE, cells can't divide properly.
Okay, so FTSE for division. What about shape, especially for rods?
That's where Mary B and his relative MBL come in. These are homologs of eukaryotic actin. They form spiral filaments just underneath the plasma membrane in rod-shaped cells. They essentially guide the machinery that synthesizes peptidoglycan, ensuring the cell elongates into a rod shape rather than just becoming spherical. If you mutate MEMB, rods become cocci.
Wow. And there's even one for curved shapes.
Yes. In curved bacteria like Caulobacter crescentus, there's cries or crescentin. It's homologous to eukaryotic intermediate filaments like lamin or keratin. It localizes along the inner curve of the cell and imposes that curvature. So you have specific proteins for division site, elongation, and curvature. It's sophisticated internal scaffolding.
Incredible. Besides the cytoskeleton, what else do we find inside? Inclusions.
Yeah, inclusions are basically storage depots or specialized compartments. You have storage inclusions for nutrients, granules of glycogen for carbon and energy, polyhydroxybutyrate or PHB, another carbon energy store, also interesting for biodegradable plastics, polyphosphate granules, storing phosphate, and sulfur globules in sulfur oxidizing bacteria.
And then there are the more complex ones, the microcompartments.
Right. These aren't membrane-bound organelles in the eukaryotic sense, but protein shells that encapsulate specific enzymes. The classic example is the carboxysome, found in many cyanobacteria and chemotaxis. It's a protein polyhedron packed with the enzyme rubisco, the key enzyme for carbon fixation. It also contains carbonic anhydrase.
Why package it like that?
It concentrates CO2 inside the shell, making rubisco much more efficient because rubisco can also mistakenly bind oxygen. The shell basically creates a high CO2 microenvironment right where it's needed. Brilliant compartmentalization without lipids.
And the really unique inclusions, gas vacuoles and magnetosomes.
Gas vacuoles are fascinating, found in many aquatic photosynthetic bacteria like cyanobacteria. They are aggregates of gas vesicles, protein structures permeable to gas but not water. By controlling the amount of gas inside, the bacteria can regulate their buoyancy and float at the optimal depth for light and nutrients.
And magnetosomes. They sound sci-fi.
They kind of are. Magnetosomes are intracellular chains of magnetic mineral crystals, usually magnetite, iron oxide, or greigite, iron sulfide. These act like tiny compass needles, allowing the bacteria to sense and align with the Earth's magnetic field.
Why would they do that?
For many aquatic bacteria living in sediments, the magnetic field lines point downwards. Following them helps the bacteria navigate away from oxygen-rich surface waters towards their preferred microaerophilic or anaerobic environments deeper down. And the precise formation of that chain of magnets is directed by another cytoskeletal protein, MAM-K structure, enables behavior again.
Amazing. Let's wrap up the internals with genetics, the nucleoid.
The nucleoid is the region within the cytoplasm where the bacterial chromosome resides. It's not membrane-bound like a eukaryotic nucleus. The chromosome itself is usually a single, large, circular molecule of double-stranded DNA.
And it's huge, right, if you stretched it out?
Oh yeah, hundreds or even thousands of times longer than the cell itself. Fitting it into the small nucleoid region is a major packaging challenge. They manage it through extensive supercoiling, twisting the DNA upon itself, and with the help of nucleoid-associated proteins, or NAPs, like the HU protein. These proteins bend, wrap, and bridge the DNA, helping to compact and organize it.
And besides the main chromosome, many bacteria also carry plasmids.
Correct. Plasmids are typically small, circular, extrachromosomal DNA molecules. They replicate independently of the chromosome and usually carry genes that aren't essential for basic survival under normal conditions, but can provide a selective advantage in certain environments, things like antibiotic resistance, virulence factors, or metabolic capabilities.
Okay, final section. Getting around and toughing it out. Motility and survival. We mentioned pili earlier for adhesion and twitching. There are also sex pili.
Right. Sex pilis are different. They're longer, fewer in number, and their job is to establish contact between two bacterial cells for conjugation, the transfer of genetic material, usually plasmid DNA, from one cell to another.
But for active swimming, we need flagella.
The bacterial flagellum is an incredible nanomachine. It's a long helical filament made of protein, flagellin, that acts like propeller. This filament is connected via flexible hook structure to the basal body, which is the complex motor embedded in the cell envelope, the plasma membrane, peptidoglycan, and outer membrane and gram-negatives.
And the assembly is interesting, isn't it? It grows from the tip.
Yeah, that's remarkable. The flagellin subunits travel up through a hollow channel inside the filament and are added onto the distal end. The basal body itself contains components very similar to a type 3 secretion system, essentially exporting the building blocks to the tip.
What actually powers the rotation of this propeller?
It's not ATP directly, it's the proton motive force, PMF, again. Protons flow through specific protein channels, the MAPOR proteins, in the basal body, moving down their electrochemical gradient from the outside to the inside. This flow of protons drives the rotation of the rotor components of the basal body, making the filament spin. It's a proton-powered rotary motor.
Amazing. And bacteria can have different arrangements of these flagellas.
Yes. Monoderm means a single flagellum, usually at one pole. Lophotrichus is a tuft or cluster of flagella at one or both ends. Amphitrichus means one flagellum at each end. And paratrichus means flagella distributed all over the surface, like in E. coli.
How does this rotation translate into directed movement, like chemotaxis? You mentioned runs and tumbles.
Right. So when paratrichous flagella, like E. coli's, rotate counterclockwise, they bundle together and propel the cell forward in a smooth run. When they rotate clockwise, the bundle flies apart, causing the cell to stop and randomly reorient a tumble.
Okay.
Chemotaxis is movement towards attractants, like nutrients, or away from repellents. The cell doesn't know where to go, but it senses changes in chemical concentrations over time as it moves. If it senses it's moving up an attractant gradient towards more food, it suppresses the tumbles.
So the runs get longer in the right direction.
Exactly. If it's going the wrong way or senses a repellent, it tumbles more frequently. The result is a biased random walk. It still moves randomly, but with longer runs biased towards the favorable direction. It gradually makes its way towards the source.
A simple but effective navigation system. Finally, the ultimate survival structure, the endospore.
Yes, the endospore, found primarily in genera like Bacillus and Clostridium. And the key thing to remember is that sporulation, forming an endospore, is a survival mechanism, not reproduction. One vegetative cell forms one endospore.
And these things are incredibly tough.
They're the most resistant known form of life. They can survive boiling, freezing, radiation, vacuum, disinfectants for years, decades, maybe centuries. This is why Clostridium botulinum spores in improperly canned food are so dangerous, or why Bacillus anthracis spores are a bioterrorism concern.
What makes them so incredibly resistant structurally?
It's a combination of factors. They have multiple protective layers. The core wall, a thick cortex made of modified peptidoglycan, a proteinaceous spore coat, and sometimes an outer exosporium. But the real keys are inside the central core.
What's special about the core?
First, it has extremely low water content, making it metabolically inert and resistant to heat and chemicals. Second, it contains high levels of calcium-dipicolinic, Ca-DPA, complexed with the DNA, which helps stabilize the DNA against heat. Third, the DNA is saturated with small acid-soluble DNA-binding proteins.
SESPS.
What do they do?
They bind tightly to the DNA, changing its conformation from the normal B form to an A form. This A-form DNA is much more resistant to damage from UV radiation, heat, and desiccation. FASPs also act as an energy source during germination.
So when conditions improve, how does it come back to life?
It's a three-stage process. Activation, often triggered by sublethal heat or certain chemicals, prepares the spore. Germination is rapid, triggered by specific nutrients. The spore swells, ruptures the coat, loses its resistance properties, and releases Ca-DPA. Outgrowth follows, where the core synthesizes new components and emerges as a vegetative cell capable of growth and division again.
What a journey, from the outer membrane to the inner workings of a spore. Let's try to boil this down. What are the, say, 3 absolute must-remember takeaways for you? Okay, first for me has to be the constant battle at the boundary. Bacteria live or die by what crosses their membranes. This drives the evolution of everything from that convoluted membrane in epilopicium to the incredible energy investment in active transport systems like ABC transporters and the PTS, just to scavenge nutrients effectively.
Good one. My second would be the cell wall as the defining feature for classification and protection. The stark difference between the thick, fortified gram-positive wall and the complex, toxic gram-negative envelope with its outer membrane and LPS dictates not just the gram-stain outcome, but also how these bacteria interact with their environment and hosts. It's fundamental architecture.
Yeah, absolutely. And third, I think, is the surprising internal complexity. We have to discard that old bag of enzymes image. The discovery of a functional cytoskeleton FETZ-AAB, core as A, organizing division and shape, plus highly structured microcompartments like the carboxysome acting as specialized factories. It shows bacteria are internally organized to a degree we didn't appreciate for a long time.
Completely agree. So building on that, here's a final thought for you, the listener, to ponder. We know that maybe only 1% of all bacterial species on Earth have actually been grown and studied in the lab. All these amazing structures and functions we've discussed are based on that tiny fraction. What completely unexpected architectural solutions or cellular machines might be waiting out there in the other 99%? What new ways of building a cell wall, moving, or surviving might we find when we finally explore that vast unknown diversity?
That's a great question. The microbial world clearly still holds a lot of secrets. It's been fantastic diving into this. Thank you for joining us for the deep dives.